Okie doke. So what did you guys come up with? I went through your discussion posts yesterday afternoon, but I didn't get to them last night. What did you guys learn? Why is science important in your life?
It's literally everywhere. Like it's just as there is math and everything, there's science and everything.
Biggest example that I could find was actually our computers that we use to meet for classes.
So technology.
Well, it's not exactly biology, but it is still a form of science.
Mhm.
I said it's important because it honestly kind of keeps humanity sane because it gives us answers for why things are, and like humans are naturally curious. So if we didn't have those answers, we would not be a good place.
Helps us to understand the universe around us. It's really important for technology. What else?
Yeah, I put it like people with like diseases and medical issues that we otherwise wouldn't be able to deal with.
Yeah, science is at the basis of modern medicine, right? Helps us to cure diseases and prevent diseases and understand how our bodies work. What else?
Well, I basically put it's like helping us being able to understand like from the universe down to the earth we live on, and like you know basically like humans where they came from, like what they are today, and like nature around us that we kind of take for granted and just walk past every morning or every afternoon.
Mhm. Yeah, so it's everything that's around us, right? Whether it's the living organisms to the geology to the study of the universe. Anything else? You guys had some really lovely posts. Good job.
And also, I think it's thinking, how we think things, real things, because we have those scientific, like, evidence there. So when something happens, you can follow those science like guidance to think things, not like in a weird way. So I think thinking also been affected by science as well.
Yeah, right. Our scientific thinking is our problem solving techniques, right? So you guys use scientific method all the time in helping to solve problems in your everyday life. It is definitely a way of thinking. All right. Well, the reason why I did this activity, we've been talking about scientific literacy this week and how important it is to understand the scientific process, right? So we're going to keep along that theme. And I wanted you guys to really reflect on why science is important, because we oftentimes forget, right? We forget how valuable science is, how at the basis of modern society it is. It is the foundation, right? The house that you live in, the infrastructure that you're using, your plumbing, your ability to drive a car, to have a car that runs, right? All that is based in science. And I think that we are in a time period where we have been undermining the value of science for quite a while. And it's kind of dangerous. And I think that what we're seeing with the COVID controversies and the misunderstanding of, how the scientific process is trying to solve this mystery of this brand new virus that we've never seen before. It's dangerous, right? It can be very dangerous to people's lives and their health, the fact that we don't understand science as a whole. So I think one of the big things that we're seeing in the last decade is a constant undercutting, right, of funding for scientific studies. Remember that science is really important for understanding how things work, right? And it's not always a direct scientific study that's going to help you to solve a problem. Again, I'm going to keep using COVID because it's topical right now. The reason why, right? So vaccines, anybody know how long does it normally take to develop a vaccine?
10, 15 years for full effect?
Can be a really long time, right? So I think average is like five years, but it can take longer. The reason why we have vaccines right now being tested is because we were able to take a lot of the knowledge that we've used from studying HIV and how that virus works and apply it to a COVID vaccine. All right. So again, science is not always I'm going to study this thing, I'm going to fund this thing, and it's going to give me this result, right? It's a process of understanding the world, right? And I think that sometimes there's misrepresentation of why it's important to fund all science, right? You never know where it's going to lead you. And so one of my favorite web pages is the Golden Goose Awards, right? So oftentimes we see people that aren't supportive of scientific funding, they poke fun at scientific studies and say, we're wasting your taxpayer money funding these studies, right? Studying bees dancing and studying the sex lives of flies. Why do we need to be wasting money on this? What the Golden Goose Awards does is it kind of talks about these random studies that you wouldn't on the face think that are important for society, and it talks about how significant they are, right? So how many of you guys use the cloud for storing music, storing data, right? Backing up your computer, keeping all your phone contacts? Anybody know where that algorithm for the cloud came from? Nobody? It is based on the honeybee waggle dance, right? So it turns out that one of the ways that honeybees communicate is by doing this little dance, right, where they bob their abdomens, and it communicates to other bees in their hive where they have found foods, right? It's how they communicate information. And it turns out scientists studying this figured out, right, how to code the honeybee dance, waggle dance, and that code is the basis of the algorithm for the cloud, right? So you never would have guessed that funding the study of honeybee dancing, right, would have been such an important study for the basis of our technology modern day. Yeah. Does this make sense? Yeah. So Golden Goose Award is kind of crazy. There's a lot of really great examples on there. So if you want to check it out, they tend to publish a lot of the ones that get brought up in Congress as, waste of taxpayer money, and show the implications. So science, pretty important to fund, right? Helps us to maintain that cutting edge on technology and medicine and, ability to survive, especially in changing climate. All right, but we'll get back to that a little bit later. So we talked about the characteristics of science. Anybody have any questions? We spend quite a bit talking about that methodological activity and why it's so important for understanding the culture and the techniques used in scientific process. Pardon me. No questions? And again, if you guys help me monitor the chat, would be great. Okay, so science is a process, right? It's a way of approaching the world. It's a way of answering questions, of exploring, of problem solving. And it's not, again, limited to life itself, right? The science application to life is biology, right? But science is a way of understanding the universe and beyond, right? And everything that it contains. So it's not just limited to life, right? All right, so what do we think? Is science the truth?
No, I don't think so, because in the biology lab, one of the discussion questions that we had was basically saying that science is never set in stone, because there's always new advances in technology and stuff and new studies. So something that's true one day could be proven to be completely different the next day.
Mm-hmm. Yeah, so that's a really great response, right? What I'm getting at here is how we were talking about science has a language of its own, right? And it's kind of different than everyday use of some of these words. So indeed, right, when we think of truth in everyday life, we think of absolutely true, or I know it in my heart, it's true, right? And that's not quite how science works, right? So, and it's not the definition that we use as truth in science, right? So the truth has many definitions, and we tend not to use this "science is the truth," right? Because it implies that it's an absolute. And in science, we don't have absolutes, right? And we'll talk about why that is in just a second, right? So in science, truth, right, is fact-based, right? It's an accumulation of facts that help us to understand the world around us. And the reason why we don't say it's the truth, right, because indeed you always are gonna have new information coming in, new study, new technologies that are gonna give us different perspectives, right? So in science, we're really cognizant of the truth, right? Or the facts are what we understand right here, right now, right? If we were yeah. So does that make sense? Right here, right now. The key thing here is that we are leaving the door open for new information that might change our understanding and our perspective. Right? But it is fact-based, and we'll talk a little bit about what's a fact versus a belief in a minute. Right? And the process of science is this process, the scientific method. So you may have talked about this a little bit in lab. You will definitely be running scientific methodology lab next week, looking at scientific method and scientific experimentation. So we'll get to that in just a bit so that you're all ready for lab next week and understand a little bit about the techniques used in the scientific process and why they're important. Any questions on, is science the truth? All right. So we said science is fact-based. What is a fact? I want to make sure that we're all on the same page, right? Because I think that there's a little bit of blurriness right now as to people's understanding of what is fact and what is not, right? A fact is information that is objective and based on evidence. It's verifiable, right? A fact is verifiable, has its basis in reality. Right? It is something that has been documented, right? And is tangibly provable, right? Verifiable, I should say, not provable. And that's very different than a belief. Right? Very different than a belief. What's a belief? How is a belief different than a fact?
A belief is when you have faith in something, but no proof is really required. So like a common example you could use would be someone's belief in a certain religion.
Yeah, a belief is a viewpoint, right? But you don't have actual factual evidence for it. So a better way to think about this is that a belief is opinion-based, right? It's just what you're thinking. And here's why I want to bring this up is because sometimes, right, when you tell yourself your belief again and again and again and again, right, we tend to adhere to that really strongly and forget that it might not be fact-based. Right? Or if you hear something that is see something that is a belief on the internet, it reinforces your belief, your opinion, right? May not necessarily be a fact. Does this make sense? Belief and facts are different, right? Are very different. All right. So getting back to that, is science the truth, right? We don't say it's the truth. I'm 100% sure this is it. Right? And this is one of those cultures of science that I think is misunderstood, right? In science, you never say you're 100% sure. You never say this is absolute. We know this to be 100% and we're not going to change our minds. Part of the culture of science is to always leave room for new information, right? Or in other words, to avoid dogmatic thinking. Dogmatic thinking or a dogma, right, is when you have a belief or an understanding of something and you are unwilling to change that viewpoint, regardless of what new data or new information comes in, right? So part of the culture of science is to always be open to new information, right? And this one's a tough one for us as humans, because our human behavior is very much of how many of you guys have had that moment with your friends or your parents or your siblings where you are proven correct. You are right and they were wrong. And you're like, I was right, right? It feels really good, right? We like that a lot, right? We don't like to admit that we're wrong, right? And sometimes that closes our ability to take in new information. Right? That's dogmatic thinking. And that's very detrimental to the scientific process, right? Because we're constantly out there gathering information. We're constantly questioning existing ideas. And we're constantly getting new technology that helps us to understand the world better, right? So culturally, again, right, leave the door open to new ideas. We try very hard in science to avoid dogmatic thinking. It doesn't mean that it doesn't happen, right? It happens sometimes. My favorite example is the 5 kingdom system. How many of you guys have heard of the 5 kingdom system? Plants, animals, fungi, bacteria, and this crazy group that we used to call Protista, right? We like it. It's one hand's worth of kingdoms, right? We still use it. We still talk about it. We still see it in books. We still see it in magazines. We still see it on the internet, even though we know the 5 kingdom system is not accurate, right? It's not taxonomically correct. We now know that the kingdom taxonomic group doesn't really stand up to genetic DNA analysis, right? And we know that if we want to keep kingdom-like groups, there's more like 30 of those groups, right? How many of you guys saw 30 kingdoms listed in your textbook? Right? We've been using that 5 kingdom system so long, right, that it's hard to let it go, right? Especially when it's 30 kingdoms. But part of that, I should actually be completely fair on that. And part of it is we're still working out those taxonomic groups that, as we're learning how to sequence DNA and clarify the diversity of life on Earth, right? So we're still working out. Maybe someday we'll see all the kingdoms listed in the textbook. Sometimes they're changing on a month-to-month basis. It's kind of crazy. Does it make sense? Dogmatic thinking? All right. So the reason why I bring this up is because we've been talking about that mistrust of the scientific process and the misunderstanding of the scientific process. And this is a big one right now. And we hear folks that say, oh, well, I don't trust those scientists because they won't say that they're 100% sure. Right? That's the nature of science. You're never going to hear a scientist say they're 100% sure. And if they do say they're 100% sure, then you know their scientific background is shady, right? We always look for 95% confidence. That's part of how we statistically analyze things, but it's also constantly reminding us to leave that window open for new information, right? Based on the information we have now, right, we will say we're very confident on an idea, but we will never say we're 100% sure, right? And you'll oftentimes see people using that to build distrust in scientists, right? That you can't believe them because they're not 100% sure. Well, it's a cultural thing in science. You can be pretty darn sure about something, but you're never going to say 100%, right? And it's not something that should be viewed as untrustworthy or the scientist not believing in their work. Right. The idea is you're leaving the door open for new information. You're open to growth and progress. Right. So I will counter you with the person that you shouldn't trust is the person that's 100% sure. Right. Because if you're 100% sure, that means that you will never listen to anyone. You will never listen to input. You will never take in new information, new data. Right. You're stymied. You're never going to progress in your thinking or in your beliefs, right? Does that make sense? Or in your knowledge base, I should say. Good? Seems silly and kind of intangible, but the stuff we're talking about today, but I think it's really important, this scientific literacy. All right, any questions? getting at culture of science. All right. So last Tuesday, we were talking about scientific method and how it's so important so that you can publish your methodology and have others assess your work and validate it by repeating your experiment. Today, we're going to talk about the scientific method, that process, right, that all scientists use across the world. It allows us to be able to share information and have it be, relatable. It allows folks to use the same approach when they're studying things. It allows other scientists to repeat your work, right? Scientific method also is really important for helping to eliminate bias and prejudice that we might have as we are exploring the world. Because of that, again, we like to be right. So sometimes that shapes our perceptions, whether consciously or unconsciously, we tend to see, right, what we want to see to reinforce our existing ideas and beliefs, right? So in science, a lot of the procedures are to kind of get around that and try and avoid that so that we have a solid knowledge foundation. Remember, the goal of science is to build knowledge and to share it. And the scientific method, again, gives you that methodology so that others can repeat your work. We said that's atypical to everyday life. You want others to repeat your work because that repeatability is going to provide you validity to your results. The more that different scientists get consistent results in an experiment, the greater confidence we have that those results are consistent with life, right? Good scientific method. All right. So before I get into the steps of the scientific method, I want to talk about the difference between a hypothesis and a theory. And again, this is one of those scientific language things. It's a little bit subtle and different than how we use these two words in everyday life. Right? In everyday life, a hypothesis and a theory are two terms that are used interchangeable. They both represent some idea that you came up with. Right? And in science, right, a hypothesis and a theory are two very different things. A scientific theory is not just a random idea, right? It is a really well-established body of work, right? And so we talked a little bit last class on how in the media, you have a lot of misrepresentation of science, and it gives people a sense that they understand science when in fact it is misinforming them. Right, and hypothesis and theory is one of those examples, right? Where in TV shows, see if you can pick it out this weekend. If you're watching any, crime solving or mystery shows, you'll see that they'll have like a, forensics scientist pick up a piece of evidence and go, "Oh, I have a theory about what happened in this crime." Right? And that's a misuse of the term theory in the scientific context, right? A tentative answer is a hypothesis. A theory is something totally different. So I want to make sure that we really understand the difference between a hypothesis and a theory. And again, in terms of misunderstanding science, we hear it all the time by folks who don't understand the language of science that, oh, evolution, it's just a theory. Those scientists aren't sure about it. Why should we teach this to our children, right? With greatly misunderstanding the fact that in science, a theory is as close to scientists saying, we're pretty darn sure about that, right? That's as close as you're ever going to get to absolutes in science is our theories, right? It is not a title that is given readily. Did we I feel like we talked about this already. No? Okay. I've given this lecture, I think, 4 times already, so I'm having lots of deja vus. All right. So hypothesis, right, is a tentative answer or explanation to a question. This is the definition of a hypothesis. How many of you guys have heard the definition? A hypothesis is an educated guess. Right. So that is a definition that we used a long time ago, right? It's a really old school definition, and sometimes you'll see people still use it. But the idea now is that, we want to start to correct the misperceptions that we're giving with the words and the language that we use, right? To say that something is a guess means it has no basis. You're just wildly throwing ideas out. And a hypothesis is not a wild, crazy idea, right? Because it is based on background information, right? So it's not a random guess. It is based in background information. You don't have to be a specialized scientist to be able to form a reasonable explanation for something based on background information. So for example, if I got into my car this morning, right, and it wouldn't start, do I have to be a mechanic to be able to formulate a reasonable hypothesis as to why my car didn't start? No, right? What do you guys have? What are some reasons that my car might not have started? You don't have to be mechanics either.
Battery.
Yes.
Battery, gas, starter. That's about as far as I can go with car knowledge.
You can look on the dash for more information.
Pardon?
You can look on the dashboard for more information even after that.
Yeah, you can have the little
Tire flat.
Well, the tire flat wouldn't necessarily cause the car not to start.
You just couldn't go anywhere.
Yeah. All right. So my point being, right, we can all form hypotheses, right, based on background information. You don't have to be an expert, right? But it is based in some knowledge, right? It's not a random guess. So tentative explanation. The other thing is that your hypothesis, a well-formed hypothesis, I'm going to drill this in a little bit, right? Because this semester you're going to be forming hypothesis in lab, and this is one of the things that we want you to be able to master. Right? A hypothesis is always going to address cause and effect. Right? A hypothesis will always include in it cause and effect. So if my car is out of gas, then it will not start. What's the cause?
No gas.
No gas. And the effect?
Your car won't start.
Your car won't start. Right. If the battery is dead, the car won't start. That has cause and effect as well. Your hypothesis should always have cause and effect. Always, always, always. All right. Your hypothesis should also be simple, reasonable explanation, logical, using critical thinking, right? Using empirical evidence. For example, I did not say the car did not start because aliens came from outer space and sabotaged me so that I could not leave the house today. That's not reasonable.
Wow.
That's not logical.
That sounds lit.
That, right, is also not testable, right? So your hypothesis has to be testable, has to be based in empirical evidence. It has to be tangible so that you can falsify it or support it. Does this make sense? All right. So say we are, putting our phones down and observing the wildlife on West Valley campus as we walk from one class to another, dreaming of a future of face-to-face classes. And as you're walking, you see a bird in the tree acting crazy. Your observation. We're going to learn our second step of the scientific method is going to be asking a question, right? Just like a reporter, who, why, what, when, where. Why is that bird acting crazy? What causes that bird to act crazy? When does that bird act crazy? Right? And your next natural step is generally to formulate a hypothesis, right? A tentative explanation. Right? Birds will act crazy when they are horny. What do you guys think of that hypothesis? What do you guys think? What does act crazy mean?
Act crazy is kind of vague.
Yeah. What about birds being horny?
And
Can-
like mating season kind of makes sense.
But can you go out and run an experiment? Hello, little birdie. On a scale of 10, could you please tell me, 1 to 10, how horny are you today? I can't do that, right? So not a valid hypothesis. Any other ideas? You don't have to be a fancy ornithologist. How could we study that?
Have like a control group and, you know, I don't know actually how you would have a control group for this scenario, but I don't know. You could just throughout the day you can test their energy levels. I guess. I don't know, man. I'm really trying right now. I don't know.
That's
It's a
good.
difficult one. You put me in a pickle.
I guess you could try to observe other birds of the same species to see if they're acting the same way.
Mhm.
That's a good one.
What causes any organism to get horny, or most organisms? Anybody know?
The time of the season for sure.
Season is a cue.
Mm-hmm.
What actually drives that behavior?
Isn't it like
Testosterone, right? It's the beauty of testosterone. It drives sex drive, right? So could you test testosterone in birds?
Yeah.
Yeah, right. So I could do something like birds with high levels of testosterone will exhibit mating behavior. Can I test that?
Yeah.
That's testable, right? And here's the cool thing about hypothesis, right? What I see frequently in lab is students are like, oh, I have to change my hypothesis. I was wrong. This is the beauty of science, right? And it feeds our liking to be right. In a in a hypothesis, right, you can never be wrong. Because when you test that hypothesis, we're going to say maybe the data is going to support my hypothesis. It shows that all the birds exhibiting mating behavior, right, had high levels of testosterone. My hypothesis is supported. Ooh, I'm on the right track. Maybe, right? I can now go through and do another set of experiments to show how testosterone is driving that behavior. However, if my hypothesis is wrong, right, all the birds exhibiting mating behavior had low testosterone levels, I can now rule, or inconsistent testosterone levels, I can now rule out testosterone as a driving force. Right? That I can take off my list, and now I go and test something different. Right? So your hypothesis gets you information, whether it tells you, oh, you're on the right track, or no, that's not the pathway, take another one. You always get information. You always win. So beautiful, right? So if your hypothesis is unsupported, right, then you test again. And again, here's a subtlety. Just because testosterone levels are high and the bird's exhibiting mating behavior, you've supported that hypothesis, but you don't say my hypothesis is true, because you don't actually know if testosterone is driving the behavior. At this point, you would just know that the two variables are correlated to each other. Birds with mating behavior tend to have high testosterone levels. You still have to show how those two are connected. Right? So again, in language of science, hypotheses we say are supported or not supported. Right? We never say they're true. Right? We just say they're supported or not. That makes sense? It's a subtlety, but you'll get more of that as you go through. Okay, so let's do a little poll and test your understanding of hypothesis. So here's the scenario, and so this scenario was given to Bio 11 human biology class. And so let me tell you the scenario, and then we're going to take a look at the hypotheses that Bio 10 or 11 students came up with, and you're going to assess them. Right? So these are going to be actual hypotheses that were formed in a lab. So your observation is that maybe you're a psychiatrist and you're treating your patients that have anxiety with this medication, drug X, right? And you notice that they all have lower blood pressure. You make this observation, not only is their anxiety reduced, their blood pressure is reduced. So your question is, I wonder if drug X is causing lower blood pressure. Yeah, valid question? Yeah. Okay, so let's take a look at some hypotheses. How do I do this? So I just tossed you a poll. Why don't you evaluate the 6 hypotheses and see what you think? Almost there, a little bit more than halfway there. So while you guys are voting, Thursdays are the day that my produce box gets delivered. So at some point, probably in the next half an hour, there will be some dog barking. I apologize. I have him tied down under the table and we'll see if we can control him.
Yay, puppies.
Naughty puppy.
Well, it's a dog. It still makes people happy.
I'm very happy. Although he might give me high blood pressure sometimes.
Yay, dogs in general.
Two more. Last call. All right. Okay. So here are our poll results. All right. So what do you guys think? That first option? Yes.
Just doesn't really. It's not like a very good It's not a formal answer whatsoever.
The first 3, 4 of the answers don't make sense out of context.
Yeah, so generally your hypothesis should stand up on its own. What's missing in those hypotheses?
There's no cause and effect on the first 4.
Yeah, there's no cause and effect, right? Drug X will work. What does that mean, work? It doesn't work. As the hypothesis. Blood pressure will be lower. How would you describe that statement? It is your getting closer to being a hypothesis. It's half a hypothesis. Let me give you that hint. Which half of the hypothesis is blood pressure will be lower?
The
Just
effect.
the effect.
Yeah, it's the effect, but the cause is missing. All right. So how about 5? If people take drug S, I think it might lower blood pressure.
You sound doubtful. Like I know we're not supposed to be 100% sure, but you should at least be confident.
What else is problematic about that hypothesis?
The I think. It makes it opinionated rather than factual.
Right. You don't want to use I think. And it's more of a prediction than a hypothesis. You're predicting what you think the outcome is going to be. All right. So great job. If you take a look at that last option, most of you are on track. If you take a look at that, it's got a cause. Or patients are taking drug X, and you've got an effect. Make sense? So when you are in lab next week, in case you're in lab before class, right, you're going to be formulating hypotheses, right? You're going to keep this in mind. When you're coming up with a hypothesis, it should be a statement, right, that addresses cause and effect. It's not opinion-based. Right? Oftentimes we see if you use an if-then statement, works well for helping you to figure out how to form hypotheses. Okay. Good? Hypotheses? Tentative explanation. All right. The opposite of a hypothesis is a scientific theory. And again, a scientific theory is an accumulation of facts that are well supported. Scientists have tested again and again and again. Remember what we said about the methodology of science and how you publish your work and you're waiting for that peer review panel, right, on that journal to critique your work and edit it and throw out, right? So it's been vetted. And then when you publish, right, other scientists can then critique your work and vet it, right? So my point here is that a scientific theory is based on experimental results, right? Data that's been collected, facts that have been accumulated that have been highly, highly, highly vetted. People have tried to critique and tear those ideas apart. These are all the ideas that float to the surface after extensive study, right? So it's an accumulation of information, right, that's shown to be really, really strongly supported. It's a comprehensive set of ideas that explains some type of natural phenomenon, right? Usually lots of scientists, lots and lots and lots of hypotheses that have been tested. Make sense? It's as good as it's going to get in science, as close to the truth as you're ever going to say, right? Can you guys give me any examples of scientific theories?
Evolution.
The theory of evolution.
Big Bang Theory.
Big Bang Theory. It's a good one. Are you going to go out and disprove Big Bang Theory on the weekend? Probably not, right?
No.
Probably not. All right. And if there's any theory that's been tried to be disproven, it is the theory of evolution. I don't think there's any other theory that has withstood as much critique and tearing apart of. So here's just a little chart with some examples of scientific theories, right? Plate tectonic theory. So that's one in geography or geology, pardon me, probably would kill me for saying that. Geology, which is the study of the lithosphere, the crust of the Earth's surface and how it's changing over time and contributing to the rock cycle. Next week, we're going to talk about this theory a little bit down here, the atomic theory. That's our understanding of atoms, right? Theory of evolution, cell theory. We're going to talk about that in a couple weeks. And theory of matter and energy, right? The idea that matter is not created or destroyed, it's just cycled. And we'll talk about those theories next week also, a little bit, or part of them anyways. Does this make sense? Theory is not the same, right, as a hypothesis. Okay. All right. Are you ready for the scientific method?
Yeah.
Yay. So scientific method isn't something that should be new to you, right? You probably use scientific method every day at some point in your life, right? So here are the steps of the scientific method that you want to be familiar with, right? And know their order. And you're going to be working with them in lab next week, and we're going to see them a few times, the steps throughout the semester. So as we mentioned earlier, right, a scientific method always begins with an observation. right? Whether it's the observation of those crazy birds or those birds acting crazy, right? Or it's the observation that, oh, the remote control is not on the coffee table. That's an observation, right? Oftentimes, always naturally leads to a question: the who, why, what, when, where. Why are those birds acting crazy? When are they acting crazy? What causes them to act crazy? Or where is the remote control? Can you formulate a hypothesis next? A tentative explanation, right? That's answering your question, right? Birds that have high levels of testosterone, right, will exhibit mating behavior. The hypothesis. Right. And then you can make a prediction based on that hypothesis, that the birds that have high testosterone levels will have lots of mating behavior. You can predict that the birds with low testosterone will have very little mating behavior. You can make a prediction. So prediction we sometimes see included in scientific methods. Sometimes it's not a step that's included. That was a weird one. But I stuck it in there because your textbook has it in there. All right. Next, you're going to run an experiment. Right? You're going to be testing that hypothesis to show either it's supported or not supported. I'm going to talk about experimental design next, so I'll come back to that. Once you run your experiment, you formulate your conclusions to assess whether your hypothesis is supported or not. If it's supported, great. Now you ask a new question. If it's not supported, you reformulate and try and get at what you're answering your question using a different hypothesis. Does this make sense, the steps of the scientific method? Yeah, pretty straightforward. Okay, so let's talk about scientific experimentation. And so normally we would run through a little mock example on the board, but I'm going to try and just like describe it because I did try and draw this, but it didn't work so well in one of my other classes. So I'm just going to try describing it to you. We'll see how that goes without a visual. Okay. So let me give you the definitions of the experimental variables, and then we're going to talk about a sample experiment, right? To kind of get you familiar with the application of these variables. This is also going to be the content of your homework assignment for the weekend. You're going to watch a video on elephant communication and a study looking at elephant communication. It goes through and it describes the processes of the scientific method that the scientists are going through, and also it goes through their experimental design. And you're going to be answering the questions of what was the observation they made, what was their question, what were the experimental variables. Make sense? And so that's due Tuesday before class. It's already up on Canvas. It should pop up as soon as class is over. And then we can talk about it a little bit more in class next week. All right, so first variable that we're going to talk about is the independent variable. And an independent variable is oftentimes described as the variable that is manipulated in your experiment, right? It is the variable that's influencing the dependent variable. It is the cause. It is the cause. What? You mean my independent variables in my hypothesis? Yes. So when you formulate a hypothesis that has cause and effect, it's setting up the independent and dependent variables that you're going to be testing in your experiment when you test that hypothesis. But I'll circle back to that in a second. All right, so that independent variable is the variable that's being manipulated. That means the dependent variable is the variable that you're interested in. It's the variable that's going to be influenced by the independent variable. So if it's things that you're interested in, right? So if I am talking about the mating behavior in birds, right? That example that we've been talking about for a little bit, what's the dependent variable?
Testosterone.
You guys agree? Everybody agree?
Agree.
Remember that testosterone drives the behavior. So do you guys think it would be the independent variable maybe? Remember the independent variable is the cause. So we think testosterone levels will drive mating behavior. And remember, we predicted high testosterone levels will cause lots of mating behavior. Low testosterone levels will cause less mating behavior. So that was a little bit hard because we haven't set up the experimental design yet, but it's the cause, right? The testosterone level would be the independent variable in that example. How about the dependent variable? It's a good guess.
Mating levels.
Yeah. How much mating behavior you see, the thing that you're interested in. Right. All right, so let's hypothetically talk about an experiment. So imagine we have that testosterone levels will cause or drive mating behaviors hypothesis. Maybe we capture a bunch of those birds and we bring them back to a lab where we're going to test, right? Different levels of testosterone and see how it affects their mating behavior. So here's what we're going to do. Here, delivery truck. We're going to remove the testes and the adrenal gland, or we're going to test males. We're going to remove all their testes. Why would we remove all their testes? What do testes do?
It produces the testosterone.
Yeah, it produces the testosterone. So we're going to remove their testes. Right?
They're removed so the scientists can control the amount of testosterone that's in their system.
Yeah. All right. So we're going to put little time release capsules in their body instead. And you're going to have capsules that release a little bit of testosterone. I'm being very scientific here. Medium levels of testosterone and high levels of testosterone. What are you guys here?
It is.
Yeah? Okay. So we're manipulating the, hey, come on. We're manipulating testosterone
Where
levels.
does Miss came?
We're manipulating testosterone levels. That's the independent variable. Could you make a prediction as to which of those treatment groups, low, medium, or high testosterone, are going to exhibit the most amount of mating behavior?
The highest group.
The high group. You got it. Make sense? Our experiment so far? All right. Now, what do you think? Is there anything else that might influence mating behavior other than testosterone?
Maybe the females present in the area.
Females present. What else?
Age.
Their age. Young whippersnapper males versus older or mature males.
The mood of the time, the mood of the time.
The time of year, right? So it could be light levels or temperature levels. Does that make sense? There's other things that could be influencing mating behavior and triggering it. So for those things, we're going to designate them as standardized variables. Right? All the other things that could influence a dependent variable, we're going to standardize them. And what that means is we're going to give equal amounts of each of those things to all of our experimental groups, right? So they will all be in a chamber that has the same temperature, that's receiving the same light, right? They'll all be the same age. We're going to standardize and hold those variables constant across our experimental groups. We're going to exclude females. So there's no influence there. Now, the goal here, standardized variables is kind of tricky. The goal here is you're guessing these other things are probably influencing mating behavior. But if I give all my experimental groups equal amounts of light, of temperature, right, age, right, then I can assume that they're all being influenced equally by the standardized variable. We're giving them what I like to call even Steven, so that they will be even influenced by those variables. Now, we can make a big assumption. Once we standardize those variables, we can assume, right, that any difference amongst the experimental groups can be attributed to the one variable, the independent variable that we're manipulating. Right? Because they should be being influenced equally when we standardize all the other things. Light, humidity, temperature, age. Does that make sense? The big assumption, right? Standardized variables.
Wait, what is the independent variable in this experiment again?
The testosterone levels.
And the dependent variable would be?
The mating behavior.
Okay.
All right. So a confounding factor is something that might go wrong. Something that you didn't take into account. Maybe we didn't realize that in the absence of females, no mating behavior would occur. And remember, we excluded females. That's something I made a mistake. So your confounding factors, sometimes you can, do secondary experiments to kind of adjust for them, or sometimes it causes you to scrap your experiment, start over, redesign. And I don't know if I told you the story where, when you go to a grad school, 90% of your time is figuring out your experimental design. 10% of your time is running the experiment and writing up your thesis. Right? The hardest part of science is designing the experiment accurately, right? So that it's actually showing what you think it's showing. Good so far? Confounding factors are the plague. Yeah. Things that you didn't take into account, things that went wrong, mistakes that were made. All right, the control group is going to be your reference. So sometimes it can be your baseline data, right? And sometimes it's a group that you're running independently to try and figure out if your design was proper. So in our bird example, here's what we're going to do, right? We're going to take naturally high testosterone males in outdoor enclosures and measure their mating behavior as a reference, as a control. Now, if we have high testosterone males in that outdoor control group, which experimental group do you think they're going to be consistent with, their behavior is going to be consistent with?
The high dosage one group.
Yeah. You would assume that a naturally high testosterone male would have pretty similar results to your high testosterone treatment group, right? So at the end of your experiment, say your high testosterone experimental group, when you compare all your data and compare them to the control group, your high testosterone experimental group exhibits 10% of the mating behavior that your control group did. What does that tell you?
Is the control group Wait, what is the control group? Like in this experiment, the birds that didn't have the testosterone added to them?
They were just naturally high testosterone.
Okay. And then you're saying like what happened, or what can we say about the experiment if the birds that did have extra testosterone added into them were like 10% more, like
10% of the exhibited 10% of the behavior that the control did.
Oh, okay.
So the control group exhibits way more mating behavior.
I don't know. I don't want to sound stupid, but like maybe it means that you like added too much testosterone to the birds.
It tells you something went wrong. So yes, you're right. Maybe you added way too much testosterone and it short-circuited them. Like, they're so stressed out they couldn't cope. It tells you something went wrong. Right? So now you have to go back and take a look at your experimental group and say, okay, what didn't I take into account?
Yeah. Okay, cool.
Maybe that surgery impacted how much energy they had for, or maybe the type of testosterone that you used in your testosterone pills was the wrong form of testosterone. It didn't replicate natural testosterone well. So the point here is your control group allows you to have a reference to identify when something goes wrong. Does that make sense? So sometimes it's used as a baseline, and sometimes it's used to help you, vet if your experiment worked.
Okay. Yeah, that makes sense.
Yeah. Okay. So I'm sorry, I should have put some like fake bird pictures in with some numbers, but coulda, woulda, shoulda. Teaching online is every day's a lesson. All right. So any questions on experimental design? Actually, before I move on. All right. So again, go back, review. If you're still not 100% clear, you can come to office hours, or you can read the text, right, to see if there are definitions and description works a little bit better. And then once you're feeling good about scientific method and scientific design, then go ahead and do that homework assignment and see if you can pick out the variables in the experiment. Right? It can be tough to think about initially. Yeah.
So I have a quick question. So for the scientific experiment, all this factor independent variable and all this 1, 2, 3, 5, they always have to be included, right? Considered, right?
Yes.
Thank you.
And I forgot to mention, I think your dependent variable is the effect, right? So again, your cause and effect and your hypothesis are your independent and dependent variables. Your hypothesis is already setting up your design. So in other words, your hypothesis tells you what the experiment is going to be. All right. Okay, doke. So once you run your experiment, you collect some data, right? We start to look for patterns in data, right, to help us see, explain what's going on in whatever we're happening to study out there, right? Those data sets can be hard to interpret. So we'll use things like statistics to analyze them to see if our experimental groups were significantly different from each other. We oftentimes use visualization. When we plot data, it helps us to identify patterns, right? It helps to convey the results of an experiment. So in this experiment, or this graph, we can tell what happened in the experiment by what's plotted in, right, on the graph. So you've got your experimental group and you've got your control group. And notice how we're testing the effectiveness of tea in relieving cold symptoms. Right? Your experimental group was receiving echinacea tea. Right? With the idea, you're testing to see if echinacea is effective at treating cold symptoms. What do you guys think the control group would have been here? What do you guys think the control group was in this experiment?
Not taking the tea.
So what do you guys think? No tea?
People who had the same sickness but were not drinking the tea. Yeah.
No tea at all?
Maybe it's
Yeah.
other type of teas. Or the
Placebo tea.
the standard of the tea like help relieve the cold.
Yeah. So, hey, what do you have? What do you have? Oh, that's fine. Tea with no echinacea. Right. Tea with no active ingredient or potential active ingredient. Why wouldn't you just give them no tea? Oh, so I think you got that. That's what you were getting at. Maybe the hot water had an influence, right, on alleviating some of the symptoms. In that case, it wouldn't be effective to have your control have no tea. That would be another independent variable that you would be adding. Right? This makes sense. The control group, tea, maybe chamomile tea with no echinacea in it. So, what would you have expected in the control group? If you're testing echinacea's effectiveness, what would you have expected in the control group in terms of effectiveness at relieving cold symptoms?
Be not as effective as the tea.
So not as effective. Would you have anticipated 2.7? What should you have seen in the control group? Let me put it that way. If echinacea is effective at alleviating cold symptoms, what should you have seen in that control group?
A lower number.
No effectiveness. If you're testing echinacea as alleviating cold symptoms, the control group that had no echinacea shouldn't have seen any relief. Right? So if your control group shows relief, what does that tell you? Did you invest in echinacea stock?
No.
No. Any ideas why that control group?
Maybe it's the hot water and not necessarily the echinacea.
Mm-hmm. Yeah, maybe it's the hot water. Right. Maybe it's the soothingness of drinking tea. So your control group helps you to interpret, right, your data. It should have been 0. All right. I don't have a lot of time to chit-chatty about this, but how do we measure effectiveness?
By the number of people that the experimental group was in. How much more how much more in the experimental group there were compared to control group?
Yeah, how much better they felt, how much relief they felt from symptoms, right? So this is in human studies, right? We oftentimes are limited because you're asking, did you, on a scale 1 to 5, how much better did you feel? Wow. Is that very rigorous? Assessing how much better you feel using a numeric scale and interpretation. What happens if your boyfriend just broke up with you? Doesn't matter how much echinacea tea you had, you're probably not going to think you feel any better, right? So when you're asking humans, right, to quantify based on opinion, not as rigorous of a study. Right? Now, if you were going in and looking at the throat and using an index of inflammation and how inflamed the throat was before and after the tea, that might be a more effective measure. So my point here is, always assess what you're basing your decisions on, right? Don't go and spend hundreds of dollars on echinacea tea. Oh, it's effective. Right? This control, look at that comparison, it tells you, I shouldn't be spending a lot of money on this supplement. It has not been shown to be really effective. I'm gonna skip that for now. All right, so our perceptions can influence results in human studies. And this is why in studies for supplements or medications, you should always have more rigorous technique involved in your experiment, right? So we oftentimes see what are called blind experiments. And this is an experiment that is trying to reduce bias, right? So a single blind study, the doctor would have, or the scientist would have given all the control and the experimental patients tea and not told them whether they had echinacea or not. So the subjects of the experiment would not have known whether they were receiving the active ingredient or not. Because you want to eliminate the, oh, I got the active ingredient, I should feel better. Oh, I'm going to tell myself I feel better. So your subjects won't know. Helps to eliminate perception bias. A double-blind study is where even the scientist doesn't know, right? So that as they're quantifying their data collection, they're not either consciously or unconsciously influenced by what they think should happen. Does that make sense? So both the researchers and the recipients don't know who's getting the medicine with the active ingredient and who's getting the sugar pill, or what we call the placebo, right? So a placebo is another fancy word that we use in medical experiments that basically means a control group. The patient's getting the tea without the echinacea, or they're getting a pill that doesn't have the blood pressure medication in it. Right. And part of why we do placebo in these types of studies is to, again, have a control, but also, interestingly enough, like we saw in the echinacea, right, control groups that aren't getting medication or aren't getting the active ingredient in the tea will sometimes exhibit the placebo effect, right, where they will have a beneficial effect. Their low blood pressure will lower, or their cold symptoms will alleviate because they think they might be getting the active ingredient. Oh, the power of your mind, right? And as it turns out, placebo effects are pretty significant. About 33% of the time, people just thinking that they're getting a medication makes them feel better or will have an effect. How crazy is that? Right. We see a lot of placebo effects in supplements, where the supplement doesn't cause any benefit, but people still feel better. So the power of your mind is pretty significant, right? This is oftentimes why when people are, being screened for experimental treatments or being screened for organ transplant, people that are in healthier states of mind are selected because they're more likely to be benefited, right? Anyhow, we already talked about graphs and figures. I'm going to mosey along because I've got 5 minutes left. Okay, so let me talk really quick about correlation and causation, right? So here's the deal with, you test your hypothesis, and let's go back to that bird hypothesis. Maybe our experiment worked and high levels of testosterone supplement, right? We saw those birds had high levels of mating behavior. Our hypothesis was supported. We've established a correlation between those two variables. When we see high mating behavior, we had high testosterone levels. That's a correlation. That initial study doesn't necessarily say, my hypothesis is true, because your hypothesis is just supported. You didn't actually, in that experiment, show that testosterone causes the mating behavior. You're the first step there. You showed the correlation, right, between testosterone and mating behavior. Your next step as a scientist would be to establish causation. You would then have to go in and study exactly how testosterone is interacting with cells in the brain to cause that behavior. You have to make the link, right? So correlation studies are always the first step to show relationships, but then you have to follow through with establishing causation, linking those two variables, right? So, for example, right? And so the reason why I talk about this is because correlational studies are great. They're always a first step. They give us information. They help us to establish patterns. But we oftentimes will read about them in the local newspaper or on the internet and think, oh, that means testosterone causes crazy mating behavior in birds. The link hasn't been shown. And until the link's shown, it's not as rigorous of an experiment. Right? And that's because it's a lot of assumption and inference that one variable is influencing the other. So let me give you an example. So there's a study done in this graphic. 2 million Americans died this year, and of those 2 million Americans that died, they were all exposed to drinking water. That's a correlation. It's true. Every single person that died was exposed to drinking water. Now, that's correlation. Does not necessarily mean that they're linked. But oftentimes we interpret that as, oh, drinking water is dangerous. Don't drink drinking water. Now, this is a silly example, but we do this all the time. We do this all the time. Supplements oftentimes show correlations, and we assume that just because there's a correlation that there's a causation. It's not necessarily the case. So when we are assessing scientific news or studies, we always want to take a look at, was this a correlational study or did they actually show causation? Right? Where are they along the process? And it's also why scientists sometimes change their mind, because an initial study showed a correlation, but they were then later unable to show the link. And until you show the causation, that link that connects them, right, it's not valid. It's just a maybe this might be influencing. Does this make sense? Right. So here's another one of my favorite examples. I don't want to spend too much time on this. Look at how tight those two lines are. Ah, those two variables must be linked. They're correlated. Right? Until you read the title. Number of people who drowned by falling in a pool in red, and Nicolas Cage movies that same year. Does that mean that Nicolas Cage movies cause people to drown in pools? There's no causation there. Could you run an experiment to show causation, potentially? It's not likely, right? But you get the idea, right? Just because two things are correlated does not mean that one's causing the other. All right. Let me wrap up here. So sample size, when you run your experimental groups, you want as big of a sample size as possible. So if you're testing that that blood pressure medication, right, your experiment that tested that medication on 100,000 people is way more reliable and valid than an experiment that tested on 100 people. Does that make sense? The more samples you have, the better. The more reliable your data is. The more reliable your patterns are. Right? And part of that is because now any outliers in your data set don't sway your values significantly. Bigger sample size is better. The other really important thing that you want to assess, right, is the randomness of the sample. You want a random sample that reflects the genetic diversity in the population of your experimental group. whether it's birds or humans, especially humans. So we see this historically in the testing of medications in the United States, where most medications early on were tested on primarily Caucasian populations. And because of that, there have been instances where, right, that medication had a different impact and effectiveness on Hispanics or Blacks. Right? So when you run an experiment, you want an experimental group that reflects the diversity in the population. And the way that we do that is by taking a random sample. Does that make sense? All right. And so I'm going to just wrap up really quickly with anti-vax movement. How many of you guys heard, oh, vaccines cause autism? So this is a great example of why scientific literacy is important and how you have to be proactive and smart about which data you listen to. Right? So this whole anti-vax movement is primarily based in one study that came out in the 90s by a doctor named Dr. Andrew Wakefield. Well, he's no longer a doctor, but Andrew Wakefield published this study. Let me tell you about it and see what you've learned. So in his study, he had 12 subjects, and 2 of the sets of subjects were siblings. So he tested on 12 kids, and all the kids had pre-existing medical conditions. So far in your analysis, what do you know about this study? What's wrong?
Incredibly small sample size.
Sample size of 12. Not reliable.
No.
The samples weren't random enough. Like you said, like 2 sets of kids were siblings.
2 sets of kids were siblings. That's problematic.
They all had pre-existing medical conditions.
Yeah, that's a huge confounding factor. Huge confounding factor.
I also did like a little research essay, and apparently the way that he got the kids in the experiment is questionable.
Yes, because their parents had hired lawyers because they wanted to sue the vaccine company.
Awesome.
So, problem with that?
There's like a conflict of interest.
Mm-hmm. An inherent bias. Not only that, and this is my favorite part. So he's a vaccine scientist, which is why people give him credit. But he had developed a rival vaccine to the most, the dominant vaccine on the market. So if he debunked and says that that vaccine causes autism, then his vaccine that he developed, that he had monetary stakes in, would become the dominant vaccine, and he stood to gain millions of dollars. Is that a problem? Yeah, huge problem, right? So he was able to publish this article in a peer-reviewed journal. So the big question is, how did he get through the peer review process? That journal never should have published that work. You guys, based on what we just learned about the study, can suss out that it was not okay. So that was a big controversy. That journal lost its reputation. And it has not gotten it back, right? That peer review process is super important. They messed up. Right? The study was almost immediately retracted. He lost his medical license over it. And yet, look at the damage that it's done. Look at the damage that it's done, right? So it's understanding the scientific process, being scientifically literate, assessing your sources is incredibly important, right? Because now you have hundreds of thousands of people worldwide Googling, right? And just because some people believe that vaccines cause autism does not mean that that belief is based in fact. Right? Subsequently to this, there has been millions of dollars in studies, right, that have been funded that have not found any causation between vaccines and autism. Right? Knowing how to assess scientific information, going through the trouble of, okay, I'm thinking of taking this supplement. Let me do some valid research to see what's the scientific basis of the effectiveness of that supplement or that medication, right? You know, also being a little bit aware of where you're pulling your sources and the authority that that source has. Right? Should you be taking medical advice from Gwyneth Paltrow? Right. All right, I'm going to stop there. Any questions? Do vaccines cause autism? That should have been my last poll.
Not likely.
No evidence for it, right? No evidence for it. Okay, let me really quickly launch my last roll poll. The variable that is manipulated in an experiment and is the cause in your hypothesis is the good job today. Awesome. So hopefully you learned a little bit about the value of scientific literacy. Hopefully we've dispelled some of the language and cultural myths about science out there. Everybody in, last call, last call, last call. Don't forget, I'm taking roll. I'm allocating points based on your roll, your poll, I mean. All right. Okie doke. Share results. So we're mostly there, right? So your variable you're manipulating, the variable that's causing, right? That's your independent variable. Dependent variable is the thing that you're interested in. It's also the effect. Right? So hopefully by the end of the weekend, you will be muttering in your sleep, cause and effect, cause and effect, cause and effect, cause and effect. Yeah. Dreaming about experimental design. So you've got a homework assignment that's due before class. Same thing, giving you an example experiment, and you're going to be picking out the variables. See if you can formulate the hypothesis that they used in the experiment. I didn't ask you that in the assignment, but we can talk about it in class. Really great job. I've got office hours in 5 minutes. If you have any questions, need any help, make sure that you're looking at the syllabus, finishing that syllabus quiz if you haven't done it already. Don't forget, you're going to have to at some point do that Proctorio assignment and do that Proctorio quiz. Get that set up well before your exam. So I'll be a little bit more forceful in getting that in closer to the first exam. But if you have time this weekend, long holiday weekend, get it done. Good. If you're not in Friday lab and you haven't already picked up your kit, go get your kits. Have a wonderful weekend.